Today we want to see about chapter 10 biotechnology and its application. So it's very important chapter. Let's get started. Biotechnology, as you would have learned from the previous chapter, essentially deals with the industrial scale production of biopharmaceuticals. The biological use of the genetically modified microbes, fungi, plants and animals. So they are genetically modified. Okay, genetically modified what? Microbes, fungi, plants, and animals. The applications of biotechnology include therapeutics, diagnostics, genetically modified crops for agriculture, processed foods, bioremediation, waste treatment, and energy production. Three critical research areas of biotechnology are providing the best catalyst. So, to provide a best catalyst in the form of improved organism, usually a microbe or pure enzyme. So, it's used to produce the best catalyst, like in the form of an organism, improved organism, usually a microbe or pure enzyme, creating optimal conditions through engineering. So, creating the optimal condition. Okay, through what? Through engineering for a catalyst to act, and downstream processes, technologies to purify the protein and organic compound. Let us now learn how human beings have used biotechnology to improve the quality of human life, especially in the field of food production and health. Biotechnological application in agriculture. Let us look at the 3 points that can be thought for increasing the food production. Agrochemical based agriculture, organic agriculture, and genetical engineered crop based agriculture. So these are the three reasons for increase the food production. The reason for the increase of food production is because of the use of agrochemical and organic agriculture and genetically engineered crop based agriculture. The green revolution succeeded in tripling the green revolution succeeded in tripling the food production, but yet it was not enough to feed the growing human population. Increased yields have partly been due to the use of improved the increased yield have partly been used due to the use of improved crop varieties, but mainly due to the use of better management practices and use of agrochemicals such as fertilizers or pesticides. So it's nothing but the increased yields have partly been used due to the use of improved, mainly have partly been due to the use of improved crop varieties, but mainly due to the use of better management practices and use of agrochemical fertilizers, pesticides. However, for farmers in the developing world, agrochemicals are often too expensive, right? So, and further increase in the yield with existing varieties are not possible using conventional breeding. As traditional breeding techniques fail to keep the pace with the demand and to provide sufficiently fast and efficient system for crop improvement, another technology called tissue culture got developed. So like if you see, farmers are facing more side effects, or they're not, it's like it's too expensive. They are finding difficulties in the by using the use of agrochemical based product because it's too expensive for them to pay for it. So yeah, so they cannot afford it, so they have created new technology that is nothing but tissue culture. developed. What does tissue culture mean? It was learned by the scientists during 1950s. At that whose that whole plant could be regenerated from explants. So all the plants could be regenerated. The regeneration will take place in the plant. That is nothing but the tissue culture says. Any part of a plant taken out and grown in a test tube under the sterile condition in special nutrient media. This capacity to generate a whole plant from any cell explant is called what? Totipotency. Totipotency. What is a totipotency? The capacity to generate a whole part from any cell or an explant. Okay, the capacity to generate the whole plant. You will learn how to accomplish this in higher classes. It is important to stress here that the nutrient medium must provide a carbon source. So here the nutrient medium must provide a carbon source, such as, like, the example of carbon sources are very important. Nothing but sucrose and also inorganic salts, vitamins, amino acids, and growth regulators such as auxins and cytokinins. So yeah, this carbon, the nutrient medium must provide the carbon such as sucrose, inorganic salts, vitamins, amino acids, and growth regulators such as auxins and cytokinins, etc. By application of these methods, it is possible to achieve a propagation of a large number of plants in very short duration, right? So this method of producing thousands of plants through tissue is called micropropagation. What does micropropagation mean? It's nothing but the production of this is nothing but the method of producing more number of plants with help of tissue culture. Production of thousands of plants with the use of tissue culture known as micropropagation. Each of these plants will be genetically identical. So all these plants will be genetically identical because they have come from the totipotency. Okay, genetically identical to the original plant from which they were grown. That is, they are soma clones. Soma clones. Soma clones means nothing but genetically identical means we can call them as soma clones. Many important food plants like tomato, banana, apple, etc. have been produced on commercial scale using this method. Trying to visit a tissue culture laboratory with your teacher to better understand and appreciate the process. Another important application of the method is the discovery of the healthy plant from diseased plant. So how to recover the diseased plant? Okay, even if the plant is infected with the viruses, the meristem, that is apical or an axillary, is free of virus. Hence, one can remove the meristem and grow it in vitro to obtain virus-free plants. Scientists have succeeded in culturing meristems of banana, sugarcane, potato. Scientists have even isolated single cell from plant and after digesting their cell wall have been able to isolate the naked protoplast surrounded by the, like, what is mean by protoplast is nothing but just surrounded by plasma membrane. Isolated protoplast from two different variety of plant, each have a desirable character and can be fused to get hybrid protoplast. Okay, so which can be further grown to form a new plant. These hybrids are called somatic hybrids. Well, the process is called somatic hybridization. Imagine a situation when a protoplast of tomato is fused with that of potato. If the protoplast of the tomato is fused with the protoplasm of the tomato, and then they are grown to form a new hybrid plants combining tomato and potato characters, well, this has been achieved resulting in the formation of pomato. Unfortunately, this plant did not have all the desired combination of characters for its commercial utilization. So here the points which we have to remember, the important points over here is, see, the scientists have isolated single cell from plant, and after digesting it by the cell wall, digesting their cell wall, they have been able to isolate the naked protoplast. Okay, and what did they do? So the same thing was performed in two different things. Okay, two different varieties of plants. Okay, two different plants, each have their desirable characters, and if they are fusing it together, they will it will result in a hybrid protoplasm or hybrid protoplast, which can be further grown. So it can be further grown into new plant. This hybrid are called nothing but the somatic hybrids. Okay. Is there any alternative path that our understanding of genetics can show so that farmers may obtain maximum yield from their field? Is there a way to minimize the use of fertilizers and chemicals so that their harmful effects on the environment are reduced? Use of genetically modified crops is a possible solution. So, what is a possible solution for this? GMO. GMO is nothing but genetically modified crops, okay, or genetically modified organisms, about which we will study over here. Plants, bacteria, fungi, and animals whose genes have been altered by manipulation are called genetically modified organisms. GM plants have been useful in many ways. Genetic modifications has made crops more tolerant. Okay, it has made the crops more tolerant to abiotic stresses such as cold, drought, salt, heat. Reduce reliance on chemical pesticides, pest resistant crops, okay, which will result in a pest resistant crops. Help to reduce post harvest losses. Increase efficiency of mineral usage. Increase the efficiency of mineral usage by plants. This prevents yearly exhaustion of fertility of soil. Enhance the nutritional value of food. Example, the golden rice, that is vitamin A enriched rice. In addition to these uses, GM, that is nothing but the genetical modification, has been used to create tailor-made plants to supply alternative resources to industries in the form of starches, fuels, and pharmaceuticals. So it's nothing but it is the genetically modified modification is also used to create the tailor-made, that is man-made plants to supply alternative sources to industries in the form of starches, okay, fuels, and pharmaceuticals. Some of the applications of biotechnology in the agriculture that you will study detail are studying detail are the production of pest resistant plants. So its application, the biotechnological applications which are used in the production of the pest resistant plants, which could decrease the amount of pesticide use pesticide used. Bt toxin. Bt toxin is nothing but the Bacillus thuringiensis. Is produced by a bacterium. It's produced by a bacterium called Bacillus thuringiensis. Bt toxin gene has been cloned from the bacteria and been expressed in plants to provide the resistance to insects without the need for insecticide. In effect, created a biopesticide. Example: BT cotton, BT corn, rice, tomato, potato, soya bean, etc. So these examples are very important. Okay, BT cotton, BT corn, rice, potato, tomato, soya bean. So now let's study about the BT cotton. Some strains of BT produce proteins that kill certain insects. Okay, so what do the BT produce? Some proteins, very important. They produce some protein to kill certain insects such as lepidopterans. Lepidopterans are nothing but the tobacco budworm, okay, army worm, and coleopterans. Okay, coleopterans are nothing but the beetles, and dipterans are nothing but the flies or mosquitoes. So what does the BT do? BT kills insects, certain kind of insects. So what are the certain kinds of insects? It's nothing but lepidopterans, coleopterans, dipterans. Okay, LCD. Lepidopterans, coleopterans, dipterans. Lepidopterans are nothing but the tobacco budworm and armyworm. Okay, lepidopterans are nothing but the tobacco worm or armyworm. Coleopterans are nothing but the beetles, and dipterans are nothing but the flies, mosquitoes. BT thuringiensis form protein crystal during a particular phase of their growth. These crystals contain a toxin insecticidal protein. Why does this toxin not kill the bacillus? Actually, the BT toxin protein exists in an inactivate protoxin, but once an insect indigests the inactive toxin, it is converted into active form of toxin due to the alkaline pH. Very important. Due to what pH? Due to alkaline pH of the gut, which solubilizes the crystal. The activated toxin binds to the surface. Okay, the activated toxin binds to the surface of the midgut. Where? Midgut epithelial cells, and creates spore that causes cell swelling and lysis, and eventually cause death of insect. Specific BT toxins genes were isolated from Bacillus thuringiensis and incorporated into the several crop plants such as cotton. The choice of genes depends upon the crop and the targeted pest. So the choice of gene, okay, the choice of gene depends upon the crop type and the targeted pest. As most BT toxins are insect specific, very important question. BT toxins are insect group specific. The toxin is encoded. There are a number of them. For example, the protein encoded by the genes Cry1AC and the proteins encoded by the genes Cry1AC, Cry2AB controls cotton bollworms. That of Cry1AB controls corn borer. Very important. Please don't confuse with this. And look at this picture. Cotton ball. Okay. A destroyed bollworm. This is what this is an destroyed bollworm, this one. Next, fully mature cotton ball. So this is what? Mature, fully mature. Pest resistant plant. Several nematodes parasite a wide variety of plants and animals, include human beings. A nematode Meloidigine incognitia infects the root of the tobacco plant and causes great reduction in yield. So, what does affect the tobacco field? The Meloidigine infects the tobacco plants. Okay, a novel strategy was adopted to prevent this infestation, which was based on the process of RNA interference. RNA takes place in all eukaryotic organisms as method of cellular defense. This method involves silencing of specific mRNA. Okay, this method involves the silencing of specific mRNA due to complementary DS. DS is nothing but the double-stranded RNA. Due to the complementary double-stranded RNA. So it's nothing but like you know that the DNA. How will the DNA look like? The DNA will look like this. But actually, how does an RNA look like? This is the structure of RNA. So this is the structure of DNA. This is the structure of RNA. So here, this is nothing but the double-stranded DNA. Why this is what? This is just a single-stranded RNA. Then how does a double-stranded RNA look like? So the double-stranded RNA also look like this only, but not exactly this, but in a slightly different form. This is the RNA. Okay, so here till this alone, it will be looking like double-stranded, but over here it will be single-stranded, but totally it is what? It is RNA only. So this is the difference between RNA, DNA, and the double-stranded RNA and double-stranded DNA. So yeah, molecule that bind to and prevents the translation of the mRNA. So what process is prevented over here? The process of translation is prevented. So they are not able to translate it. That's why translation of mRNA, and this process is called as silencing. The source of this complementary RNA could be from an infection. Okay, infection by viruses having RNA genomes or mobile genetic elements known as transposons that replicate via an RNA intermediate. So yeah, I'll just explain it once again. See, till this we are completed. Now let's start from this. The source of this complementary RNA will be from the complementary RNA will be from what? Will be from an infected virus only, and that virus should have RNA genome or a mobile genetic element or transposons, so that it can replicate via RNA intermediate. So they require a virus so that it should have an RNA genome. Using agrobacterium vector, nematode specific genes were introduced. So using what? Using agrobacterium vector, nematode specific genes were introduced into the host plants. Okay, the introduction of DNA was such that it produced both sense and antisense. So, as we introduce the DNA, it should produce both sense means let's take it as positive. Okay, it should produce both positive, they should produce both positive and negative means antisense. Okay, RNA in the host cells. These two RNAs being complementary to each other formed a double stranded RNA that initiates in RNAi. Okay, so what initiates this is this sense and antisense only initiates the process of RNAi and making the strand a double stranded and double strand RNA that initiates RNAi. And thus silence the specific mRNA of the nematode. The consequence was that the parasite could not survive in transgenic condition. The consequence was the parasite could not survive in a transgenic host expressing specific interfering RNA. The transgenic plant therefore got itself protected from the parasite. So the transgenic plant was then got protected by itself from the parasite. So yeah, this one is very important. The host plant generated double stranded RNA triggers the protection against the nematode specific nematode infestation. Was shown over here. Roots of a typical control plants. This control plant. Transgenic plant roots 5 days after deliberate infection of nematode but protected through novel mechanism. So the first one is nothing but the controlled plant. It's totally controlled. And this is what it was left for 5 days and it was protected. Next, biotechnological applications in medicine. The recombinant DNA technological process have made immense impact in the area of healthcare by enabling the mass production of safe and more effective therapeutic drug. Further, and recombinant therapeutics do not induce unwanted immunological responses as is common in in case of similar products isolated from non-human sources. So the unwanted immunological responses as is common in case of similar products isolated from non-human sources. At present, about 30 recombinant therapeutics have been approved for human use. So at present, this is very important MCQ question. At present, about 30 recombinant therapeutics is approved for human use in the world over. In India, 12 of these are presently being marketed. So out of 30, we are marketing 12 of the recombinant therapeutics. Genetically engineered insulin. Management of adult onset diabetic is possible by taking insulin at regular time interval. What would a diabetic patient do if enough human insulin was not available. If you discuss this, you would soon realize that one would have isolate the use insulin from other animals. Would the insulin isolated from other animals be just as effective as it's secreted by the human body itself, and would not elect an immune response in the human body? Now, imagine if bacterium were available. Imagine that bacterium were available that could make human insulin. Suddenly, a whole process becomes so simple. You can easily grow a large quantity of the bacteria and make as much insulin as you need. Think about whether insulin can be orally administered to diabetic people or not. Why? Insulin used for diabetes was earlier extracted from pancreas. See, the insulin for diabetes was earlier extracted from the pancreas of the slaughter cattle, pigs and pigs. Insulin from an animal source through caused some see insulin from an animal source though caused some patients to develop allergy or other type of reactions to foreign protein. Insulin consists of two short polypeptide chain. This is very important. Okay, insulin consists of two polypeptide chain A, chain B are linked together by disulfide bridges. So yeah, here insulin is made up of two chain A chain B. Okay, and they are linked with the help of disulfide bridges or bisulfide bridges. In mammals, including humans, insulin is synthesized as a prohormone. Like proenzyme, the prohormone also needed to be processed before it comes to a fully mature and functional hormone, which contains an extra stretch called C-peptide. So, we can say that we are having chain A, chain B, and also now we have studied that they have chain C. But all these are what? They are nothing but polypeptides. These all are nothing but polypeptide. This C peptide is not present in a mature insulin. Okay, it's not present in a mature insulin and is removed during the maturation into insulin. The main challenge for production of insulin using rDNA techniques. So, the main challenge is nothing but the main challenge of production of insulin using rDNA technique, nothing but recombinant DNA technique, was getting insulin assembled into a mature form. In 1983, Eli Lilly, an American company, prepared two DNA sequences. They prepared two DNA sequences corresponding to A and B chains of human insulin and introduced them in plasmids of E. coli to produce insulin chains. Chain A and B were produced separately, extracted and combined by creating disulfide bonds to form human insulin. So this will be in the form of matured form. It will be in the matured form. Gene therapy. If a person is born with a hereditary disease, uh, can a corrective therapy be taken for such a disease? Gene therapy is an attempt to do this. Gene therapy is a collection of methods that allows the correction of a gene defect. So, gene therapy is a collection of methods that allow the correction of a gene defect that has been diagnosed in the child or embryo. Here, genes are inserted into a person's cell and tissues to treat a disease. Correction of a genetic defect involves delivery of a normal gene into the individual or embryo to take over the function of and compensate for the non-functional gene. They have to compensate or they have to take over the function. The first clinical gene therapy, the first clinical gene therapy was given in 1990 to a 4-year-old girl with adenosine deaminase ADA, adenosine deaminase deficiency. This enzyme is crucial for the immune system to function. The disorder is caused due to the deletion of the gene for adenosine deaminase. In some children, ADA deficiency can be cured by bone marrow transplantation. In others, it can be treated by enzyme replacement therapy, in which the functional DNA is given to the patient by injection. But the problem with both these approaches that they are not completely curative. As a first step towards gene therapy, lymphocytes from the blood of the patient are grown in a tissue outside the body. A functional ADA cDNA using retroviral vector. A functional ADA cDNA is then introduced into these lymphocytes, which are subsequently returned to the patient. However, as these cells are not immortal, the patient requires periodic infusion of such genetically engineered lymphocytes. However, if the gene isolated from marrow cells producing ADA is introduced into cells at early embryonic stage, it could be a permanent cure. So, for a permanent cure, the early detection and early treatment will be the best thing, for a permanent cure. But as a person is grown, after which we can only just add some gene. We can just modify the gene, but we cannot completely modify the gene totally. So we have to again and again we have to do the infusion of the specific enzyme like adenosine deaminase. Molecular diagnosis. You know that for effective treatment of disease, early diagnosis and understanding its pathophysiology is very important. Using conventional methods of diagnosis, serum, urine analysis, etc., early detection is not possible. Recombinant DNA technology, polymerase chain reaction, PCR, and enzyme linked immunosorbent assay, ELISA, enzyme linked immunosorbent assay, are some of the techniques that serves the purpose of early diagnosis. Presence of pathogen, bacteria, virus, etc. is normally suspected only when the pathogen produce a disease symptom. By this time, the concentration of the pathogen is already very high in the body. However, very low concentration of the bacteria or virus. So the concentration of the pathogen will be high and the concentration of the bacteria or the virus will be low at the time when the symptom of the disease are not yet visible. Can be detected by amplification of their nucleic acid by PCR, and it can be detected by the amplification of their nucleic acid with the help of the method called PCR. Can you explain how PCR can detect very low amount of DNA? PCR is now routinely used to detect HIV in suspected AIDS patient. It is being used to detect the mutations in genes in suspected cancer patients too. So it's used to detect the mutations, what kind of mutation takes place in the gene in the suspected cancer patients. Okay, what a PCR is used over here. It is a powerful technique to identify many other genetic disorders. So PCR is used to detect all this. Okay, a single stranded DNA or RNA tagged with a radioactive molecule probe is allowed to hybridize to its complementary DNA in a clone of cells, followed by detection using radiography. The clone having the mutated gene will not appear in the photographic film because it will have different kind of pattern in their strand, because the probe will not have complementary with the mutated gene. So we will not have any complementary to the mutated gene. So during the radiography, during autoradiography, they will not have the complementary strand, so that we can detect the mutated gene. ELISA is based on the principle of ELISA is based on the principle of antigen antibody interaction. Infection by the pathogen can be detected by the presence of antigen. So the infection of the pathogen can be detected by the presence of antigen. Antigens are nothing but the proteins or the glycoproteins, or by detecting the antibody synthesis against the pathogen. Transgenic animals. Animals that have had their DNA manipulated to possess and express an extra foreign gene are known as transgenic animals. So, can you define what is transgenic? What is transgenic animal is? It's nothing but whose DNA gets manipulated. Okay, whose DNA have been manipulated to possess some extra foreign DNA molecule or a gene. Okay, transgenic rats, rabbits, pigs, sheep, cows, and fishes have been produced. Although over 95% of all existing transgenic animals are mice. So mice, the number of percentage of transgenic mice is nothing but 95 percentage. Why are these animals being produced? How can man benefit from such modification? Let us try and explore some of the common reasons. Normal physiological and development. Okay, why we are using all the such transgenic animals? Okay, we are going to study about all that. For number one, it's just to for the normal physiology and development. Transgenic animals can be specifically designed to allow the study of how genes are regulated. Nothing but to allow the study, okay, to allow the study how genes are regulated. So whatever I'm highlighting, it's the important points. To allow the study of how the genes are regulated and how they affect, okay, how they affect the normal functions of the body and its development. For example, study of complex factors involving in the growth. For this, the example is nothing but the insulin like growth factor by introducing genes from other species that alter the formation of this factor. Okay, just remember the example is insulin. We have just taken the example of insulin like growth factor by introducing these genes from other species that alter the formation of the factor, and studying the biological effects that results in the information is obtained about the biological role of the factor in the body. So just for normal physiology and development, just remember the story of insulin. Study of disease. Study of disease. Many transgenic animals are designed to increase our understanding of how genes contribute to the development of the. So why the transgenic animals are made? To increase our understanding of how the gene contribute to the development of the disease. These are specially made to serve as models for human diseases, so that the investigations they serve as the models for human being. They serve as models for human being. This is so that the investigation of the new treatment for the disease is made possible. Today, transgenic models exist of many human diseases. Okay, example: cancer, cystic fibrosis, rheumatoid arthritis, Alzheimer. CCRA. Just remember C square RA. First remember like CRA, then C square RA. Next, biological products. Okay, medicines required to treat certain human diseases can contain biological products. Okay, to treat some disease we need some biological products, but such products are often expensive to make. Transgenic animals that produce useful biological products, transgenic. Okay, the transgenic animals that produce useful biological products can be created by introduction of the portion of gene, nothing but rDNA technology of DNA, which encodes for a particular product as such. Human protein alpha-1 antitrypsin is used to treat emphysema. This is very important. Alpha-1 antitrypsin is used to treat emphysema. Similar attempts are being made to treat for the treatment of the PKU, nothing but phenylketonuria, and cystic fibrosis. In 1997, the first transgenic cow was made. In 1997, the first transgenic cow was made. Its name is Rosie. Produced human protein enriched milk 2.4 grams per liter. Here the important thing to remember is it produces 2.4 grams per liter, GL. Okay, don't forget this, very important. You may get confused with the options. 2.4 grams per liter. The milk contained the human alpha lactalbumin and was nutritionally a more balanced product for human babies than natural cow milk. Vaccine safety. The transgenic mice are being developed for use in the testing the safety of vaccines before they are used on humans. Transgenic mice are being used to test the safety of the polio vaccine. So, which animal was used to test the polio vaccine? It's nothing but mice was used to check the polio, check the safety of the polio vaccine. If successful and found to be reliable, they could replace the use of the monkeys to test the safety of batches of vaccine. Chemical safety testing. What is chemical safety testing? This is known as the toxicity of the safety testing. The procedure is same as that used for testing toxicity of the drugs. To test the toxicity of the drugs, we are using transgenic animals are made that they carry the genes which make them more sensitive towards the toxic. This is very important thing. They are more sensitive towards the toxic substance than non-transgenic animals. This one they can ask directly. Transgenic animals are more sensitive towards the toxic substance than non-transgenic animals. They are exposed to the toxic substance and the effects studied toxicity. Testing in such animals will allow to obtain the results in less time. Ethical issues. So before ethical issues, I'll just sum it up. So there are totally only 5 points. What are the uses of the transgenic animals? For the normal physiology, just remember about this insulin story. Then study of disease is nothing but a CRA, C square RA, and many transgenic animals are used for this process to study the effect of the disease on its body. Next, biological products. We can make so many biological products with the help of the transgenic animals, like making the cRNA, rRNA technology, recombinant DNA technology into the recombinant transgenic animals, so that we can produce human enriched milk, and we can also produce some alpha 1 antitrypsin, and it is used to treat the emphysema, and a similar effect is also here. Attempts are made to treat for the treatment of the PKU and the cystic fibrosis. And in 1997, the first transgenic cow was made named Rosie, and it helps in the production of the human protein enriched milk, that is 2.4 gram per liter. Then the milk contained the human alpha lactalbumin, was nutritionally more balanced than a product for the human babies than the natural cow milk. Then the vaccine safety. Yes, the transgenic maize are used for the safety test of the polio vaccine. And next one is the chemical safety testing, is also known as the toxicity or the safety testing. The procedure, like here nothing but the transgenic animals are made more sensitive towards the toxic substance than the non-transgenic substance, so that they can get the result in very less time. Next one is the ethical issue. The manipulation of the living organism by the human race cannot go on any further without regulation. Some ethical standards are required to evaluate the morality of the all human activity that might help or harm living organisms. So there are some ethical standards that everyone need to keep in their mind. Going beyond the morality of such issues, biological signification of such things is also important. Genetic modification of GMO of organism can have unpredictable results. So this is also a very important question. The GMO can have predictable results or unpredictable results. So yeah, of course, it gives unpredictable results when such organisms are introduced in the ecosystem. Therefore, the Indian government has set up an organization known as GEAC. This full form is also very important. GEAC. GEAC in full form, Genetic Engineering Approval Committee, which will make the decision regarding the validity of the GM research and the safety of introducing the GMO for public services. The the modification or the usage of the living organism for public serves as a medicine source for example, has also created problems with the patent guarantee. So some more problems gets arise when we are introducing a GMO into the ecosystem. For example, the patent rights and etc etc are the following problems for this. There is growing public anger that certain companies are being granted patents for products and technologies that make use of the genetic material, plants and other biological resources that have longer been identified, developed and used by farmers and indigenous people of a specific region or country. So we are going to study some example for this. Nothing much. Rice is an important food grain, as we know. The presence of which goes back thousands of years in Asia's agricultural history. There is an estimated how many varieties of rice was there in India. There were around 2 lakh varieties of rice in India alone. The diversity of rice in India is one of the richest in the world. India has the richest variety of rice in the world. Next, basmati rice is another story. Basmati rice is distinct for its unique aroma and flavor, and 27 documented varieties of basmati are known in India. How many? How many? 27 documented varieties of basmati rice are grown in India. There is a reference to basmati in Asian text, folklore, and poetry, and it has been grown for centuries. In 1997, what happened? In 1997, an American company got its patent rights on basmati rice through the US Patent Trademark Office. This allowed the company to sell a new variety of basmati rice in the US and abroad. It's a vast variety of the basmati which was grown in India. This new variety of basmati had actually been derived from Indian farmers variety. Indian basmati was crossed with semi-dwarf. Okay, I told you, dwarf variety, semi-dwarf varieties, and claimed as an invention of the novelty. The patent extends to the functional equivalent, implying that other people selling basmati rice could be restricted by the patent. Okay, other people selling the basmati rice could be restricted by the patent. Several attempts have also been made to patent uses, products, and process based on Indian traditional herbal medicine. Example, turmeric, neem. If we are not vigilant and we do not immediately counter these patent applications, other countries or the individuals may encash our rich legacy and we may not be able to do anything about it. So biopiracy is a term. This is very important. Biopiracy is a term which is referred to the use of the bioresources by multinational company, MNCs, and other organizations without the proper authorization from the countries and the people concerned with the compensation payment. Without giving the payment, the using of the bioresource by MNCs or other organizations is known as a biopiracy. Most of the industrialized nations are rich financially but poor in biodiversity, a traditional knowledge. In contrast, the developing and underdeveloped world is rich in contrast, what happens? In contrast, the developing and the underdeveloped world is rich in biodiversity and traditional knowledge but related to bioresources. The traditional knowledge related to the bioresources can be exploited to develop modern applications and can also be used to save our time, effort, and expenditure during their commercialization. There has been growing realization of the injustice, inadequate compensation, and benefit sharing between the developed and the developing countries. Therefore, some nations are developing laws. Some nations are developing laws to prevent such unauthorized exploitation of their bioresources and traditional knowledge. The Indian Parliament has recently cleared the Second Amendment. Okay, Indian Parliament has cleared the Second Amendment. How much? Like first or second or third? Is on the Second Amendment of the Indian Patent Bill. About Indian Patent Bill that takes such issues into consideration, including the patent terms of emergency provisions, research and development initiatives.